Hello foodies, welcome back to FoodEdge. So today in this video we are going to see about the monosaccharide reactions, and it is the continuation of the carbohydrates part that we are dealing from the food chemistry. So moving on to our first reaction, it is the oxidation to aldonic acids and aldonolactones. So aldoses are oxidized to aldonic acids, and during this oxidation, the aldehydo group or the functional group of aldoses, which is nothing but the CHO group, is converted to a carboxyl or an carboxylate group. So carboxylate is nothing but the acidic group, that is the COOH group. So this reaction is used for the quantitative determination of sugars. So the reagents used in this reaction includes the Fehling solution, Nelson-Somogyi reagent, and the Benedict's reagent. So the process includes the aldehydo group of an aldoses is oxidized to the salt of a carboxylic acid group. So the sugar reduces the oxidizing agent. Thus we call that sugar as a reducing sugar. So for better understanding, let us take the reagent to be a Fehling solution. So this reagent is going to act as the oxidizing agent. So the alkaline solution of copper 2. So copper 2 is nothing but the cupric oxide, and this is the oxidizing agent when we consider Fehling solution. So this copper 2 oxidizes an aldose to an aldonate. And in this process, the copper 2 is reduced to copper 1. So copper 1 is nothing but cuprous oxide. So this conversion of copper 2 to copper 1 is being carried out by the aldose sugar, and thus we call this aldose sugar as the reducing sugar. So with this cuprous oxide or the copper 1 precipitates as a brick red color precipitant. So the amount of the precipitate obtained at the end of the reaction indicates the amount of sugar actually present in the given sample, or the amount of sugar that has been taking part in the reaction actively. So it by this way it determines the quantitative determination, that is it determines the quantitative or the quantity of sugar present in the given solution. So moving on to the next, ketoses are also termed as reducing sugars because under alkaline conditions of the Fehling's test. So Fehling's test is nothing but an it has cupric oxide as the initial reagent, that is copper 2, which is alkaline in condition. So under this alkaline state, the ketoses are isomerized to aldoses. So we know that fructose and glucose are structural isomers of each other. So whenever an alkaline condition prevails in a reaction, the ketoses are converted or isomerized to aldoses. So, but in the case of Benedict's reagent, it is not an alkaline reagent. So it will react only with the aldose sugars, but not with the ketose sugars. So here, when the reaction is taking place in the presence of Benedict reagent, the ketoses are not isomerized to aldoses, and thus they do not take part in the oxidation reaction. So other variations like the Nelson-Somogyi and the Benedict's reagent are still used for determining the amount of reducing sugars present in foods and other biological materials. So an example for this oxidation reaction is the conversion of D-glucose to D-gluconic acid. So the reaction is commonly employed to measure the amount of D-glucose in foods and other biological material including blood and urine. So D-gluconic acid is a natural constituent of fruit juices and honey. So moving on to the next, you can see the reaction here, that is the conversion of D-glucose to its corresponding aldonate, that is D-gluconate. So here you can see the open chain structure of D-glucose, which to the next image is the ring or the pyranose form of D-glucose. So this pyranose ring structure is reacting with the oxygen, that is it is undergoing oxidation at the C1 position, that is the carbon 1 is the aldehydic carbon. So C1 position is being oxidized, and this oxygen is cleaved from the hydrogen peroxide, that is hydrogen peroxide is cleaved in order to release oxygen, and that oxygen takes place in the oxidizing of glucopyranose. So here the immediate product formed is a lactone, that is the D-glucano 1,5 lactone, And on protonation of this lactone, the product formed is D-gluconate. So this reaction is the commercial reaction to produce D-gluconic acid and its lactone. So the lactone, that is the D-glucano delta lactone GDL, otherwise it is called as D-glucano 1,5 lactone, is produced by slow acidification, and its mild taste make it unique among food acidulants. So the GDL is used in meat and dairy products as acidulants, but particularly in the refrigerated dough as chemical leavening compound. So moving on to the next is the reduction of carbonyl groups. So reduction is nothing but addition of hydrogen and also removal of oxygen. So the reduction or the addition of hydrogen, otherwise you can call as hydrogenation also. So here the reduction is taking place in the carbonyl groups. That is, carbonyl groups is nothing but the functional groups of ketosis and aldoses sugars, which is the CHO or the C double bond O. So this carbonyl group is being reduced, that is hydrogen atom is being added to this carbonyl groups. So the addition of hydrogen to break the double bond between the oxygen atom and carbon atom of the carbonyl group of aldoses or ketosis. So this is what you call as reduction. So when we consider D-glucose, the hydrogenation or reduction of D-glucose in the presence of hydrogen gas under pressure, and the catalyst used is the Raney nickel. So this is the similar setup that is being followed for the hydrogenation of fats and oils also. So on hydrogenation of D-glucose, the product formed is glucitol, which is commonly called as sorbitol, or commercial name is sorbitol. So from the suffix OL, it is very clear that they are alcohol group, and they are also called as sugar alcohols or alditols, as they are derived from the aldose sugars. So they are known as the polyols or polyhydroxy alcohols. So since glucose is a hexose, as we all know, we can also call an overall term for the sugar alcohols as hexitols. So sorbitol or D-glucitol obtained from glucose is widely distributed in plants ranging from algae to higher plants, and it is also found in fruits and berries of the plants. So it is half as sweet as sucrose, and it is sold both as syrup and crystals. It is a general humectant. So humectant is nothing but the compound added to any food product in order to retain moisture in it. So moving on to the next. So D-glucose yields D-glucitol or the sorbitol, which is commercially as it is called. From the hydrogenation of D-mannose, the product obtained is D-mannitol. So mannitol is commercially obtained along with sorbitol from hydrogenolysis of sucrose. So we all know that sucrose is a disaccharide, and it is a combination of glucose and fructose. So on hydrogenation or hydrogenolysis, as it is said, is carried out, the product formed is sorbitol and mannitol. So it is the product of hydrogenation of the fructose component of sucrose and from isomerization of D-glucose. So if you remember the last reaction where we saw that under alkaline conditions, ketosis or the fructose can be converted to its corresponding aldose sugar, that is the glucose. So, whenever we have a controlled alkalinity, we can vice versa the reaction, that is aldoses can be isomerized to ketosis. So in that case, this D-fructose component of sucrose is mainly involved in producing the sorbitol and mannitol. So D-mannitol, unlike sorbitol, is not a humectant. Rather, it crystallizes easily and is only moderately soluble. So the main use of D-mannitol is for the non-sticky coatings on candies, and it is 65% as sweet as sucrose, and it is used in sugar-free chocolates, pressed mints, cough drops, and also hard and soft candies. So here is the reaction where you can see the D-fructose part from the sucrose is obtained, and it is being reduced, that is hydrogen atom is added to the functional group. So we saw that reduction is breaking the double bond and adding hydrogen to it. So hydrogen is added to the functional group of fructose, where the first compound obtained, you can see it is the glucitol, that is Glucitol and mannitol differ only at the arrangement of the C2 carbon, that is OH is on the right and in the other thing the OH is on the left. So they both are actually epimers, if you remember from our first introduction video to isomerization of monosaccharides. So moving on to the next is the sugar alcohol, that is xylitol, and it is produced from hydrogenation of D-xylose. So xylose is a pentose sugar and it is the key building block or the primary building block of the polysaccharide hemicellulose. So this hemicellulose is mainly found in the cells of plants and especially from the bush trees where they are abundant. So its crystals, that is the crystals of xylitol, have high negative heat of solution, that is they take part very effectively in the endothermic reactions. So this endothermic heat of solution of crystalline xylitol produces a cooling effect in the mouth. So when we eat mint chocolates like the Polo or some other chewing gums, you can very well feel a cooling effect in your mouth, and this is because of the endothermic reaction that the sugar actually present in that chocolate is carrying out, and xylitol is one such kind of sugar. So this cooling effect of xylitol makes it desirable an ingredient in mint candies and also in sugarless chewing gums. So its sweetness is equal to that of sucrose. So xylitol is a non-cariogenic sugar. That is what is cariogen means. It causes dental problems like dental caries or dental plaques. And few examples of cariogenic sugars are the glucose and sucrose, which we commonly eat in our day-to-day life. So why xylitol is non-cariogen? Because it is not metabolized or it cannot be broken down by the microflora or the microorganisms that are present in our mouth, and they produce dental plaques. So since they cannot be metabolized, this xylitol on consumption will not produce any dental plaques. So moving on to the next is the next reaction. It is the formation of uronic acids. So the terminal carbon atom of monosaccharide unit of an oligo or polysaccharide may occur in an oxidized or form, that is the carboxylic acid form. So the terminal carbon atom may be the C6 of a monosaccharide unit. So we all know that oligo and polysaccharides have building blocks of monosaccharides. So in the oligo and polysaccharide sugars, if the monomer units have the oxidized terminal carbon atom, they form the uronic acids. So such aldohexoses with C6, that is the terminal carbon atom, in the form of carboxylic acid group, we call them as sugar acids or uronic acid. So on reduction of the sugars, what we get are sugar alcohols, and on oxidation of the terminal carbon atom, that is the C6 of the sugars, we get sugar acids. So two different terms which we have to know here is oxidation of terminal aldehyde yields aldonic acid. So if you remember the first reaction, it is the oxidation of the aldehydic group or the functional group of the acids, and one example we saw is conversion of D-glucose to gluconic acid, which is nothing but an aldonic acid. So oxidation of terminal aldehyde alone yields aldonic acid, while oxidation of both terminal hydroxyl group and aldehyde, that is the C1 and C6 of the aldose sugar, yields an alderic acid. So it is two different terms which we have to note. And the oxidation of terminal carbon atom alone yields uronic acid. So there are three different terms which we have to know. So oxidation of aldehyde, terminal aldehyde alone yields aldonic acid, whereas oxidation of both terminal aldehydic group and terminal hydroxyl group yields alderic acid, and oxidation of terminal hydroxyl or the last C6 carbon alone yields uronic acid. So when the chiral atoms of the uronic acid are in same configuration as they are in the sugars, they form sugar acids. That is, from D-galactose, the acid obtained is D-galactouronic acid, and it is the principal component of pectin. From D-glucose, the acid obtained is D-glucuronic acid, and it is present in gums like Arabic gum and xanthan gums. So moving on to the next. D-glucose and D-mannose, the acid produced from them is glyuronic acid and mannuronic acid. So these acids are in combination the principal compound present in the alginic acid. That is, alginic acid or algin is a polysaccharide. It is a which is abundantly found in brown algae. So mucic acid or galacteric acid, so we saw, is an aldaric acid. Aldaric acid is obtained on oxidation of both aldehyde and terminal carbon or the hydroxyl group. So this galacteric acid is obtained by nitric acid oxidation of galactose or galactose containing compounds like lactose and gums, etc. So that is all about uronic acids. So moving on to the next is other reactions taking place in the carbohydrates or the monosaccharides in particular. So it is the hydroxyl group esters, where you can see the hydroxyl group of carbohydrates, like the hydroxyl groups of simple alcohols, form esters with organic and some inorganic acids. So few examples are acetate, succinate half esters, and other carboxylic acid esters of carbohydrates occurring in nature. They are especially found as components of polysaccharides. For example, potato starch contains a small percentage of phosphate ester groups. Other esters of starch are acetate, succinate, and substituted succinate half esters and distarch adipates that are found in the modified starches. So moving on to the next, sucrose. Sucrose fatty acid esters are produced commercially as water and oil emulsifiers. The family of red seaweeds polysaccharides, which includes the carrageenans, contain sulfate groups, that is half esters of sulfuric acid. So this is what about the reaction of between carbohydrates and esters, where mainly polysaccharide exhibits the presence of sugars in esterified form. So next one is the hydroxyl group ethers, where you can see the hydroxyl group of carbohydrates can form ethers as well, but they are not very common in nature as esters. However, polysaccharides are etherified commercially to modify their properties and make them more useful. So this is what is happening in the modified starch, which we will be discussing when we are seeing the starch part. So examples, the production of methyl sodium carboxymethyl and hydroxypropyl ethers of cellulose and hydroxypropyl ethers of starch, which are approved for food use. So this hydroxypropyl ether of starch is a modified starch and which is approved for food use. So a family of non-ionic surfactants based on sorbitol, that is nothing but the D-glucitol, are used in foods as water and oil emulsifiers and also as defoamers. They are produced by esterification of sorbitol with fatty acid. The products are known as sorbitian esters. So the sorbitian fatty acid esters such as sorbitian monostearate, sorbitian monolaurate, and sorbitian monooleate are sometimes modified by reaction with ethylene oxide to produce so-called ethoxylated sorbitian esters called tweens. So tweens are the commercial names of the sorbitian esters that are produced on reaction with ethylene oxide. So they are non-ionic detergents approved by US FDA for food use. So they are used as food detergents and they are also approved by the FDA. So that is all about what we are going to see, what we saw about the reactions in monosaccharide. So we will be discussing more about the disaccharide and polysaccharides in our upcoming video. Until then, stay tuned, know and learn more about food technology. Keep watching. Thank you.